So my name is Chee Yeun Chung, and I'm a senior research scientist at Whitehead Institute in Cambridge. The thing I do is to study neurodegenerative diseases like Parkinson's disease or Alzheimer's disease using a model system. I was born and grew up in Seoul, Korea. So I actually am the first scientist in the family. This mentality of trying to make things better and understand how things work, I think definitely helped me going into science and also do well in science as well. I did my undergraduate in Seoul College of Pharmacy, but I ended up studying neuroscience actually, because I always had a fascination about brain and neurons and why is human being a human? As the population grow older, there are more people suffering from Alzheimer's disease, Parkinson's disease. There are many other neurodegenerative diseases because these are diseases of aging. So the burden to the society has been very grave, and people now, at the government level, people are trying to tackle this problem. Sadly, there's no disease-modifying therapy in neurodegenerative diseases. What we are trying to do here is to marry the simple model system like yeast, that is a unicellular organism, and with a much more complex model system like neuron system. Discovery of this technology called reprogramming. Now we can get neurons from your brain. Now we can get embryonic stem cell-like cells from your body and my body. To get the embryonic stem cell-like cells from skin cell, it was like a magic. We call it now pluripotent stem cell, iPS cells. We can redifferentiate iPS cells into many cells in your body, including neurons in the brain. So what I'm doing is these are gels that you can put your sample in. This is called power supply. This power supply is now providing positive charge and negative charge. And as I said, protein has many different sizes, and that's going to have different charge depending on the size of the protein. And what we try to do in this paper of ours is that we wanted to compare whether we can find anything going wrong in these cells. But the cells are so complex. It's like a navigating jungle. It's so hard to navigate or where we are going and what is actually going on in these cells. So we thought that maybe we can use a very simple cell system that has conserved biology like human, but it's just so simple that we can do unbiased screening. Things that we found in yeast cells, the simple model system, also can be replicated in human neurons. And we can use this information from simple cell like yeast to navigate in this complex cell system. But we can also get an idea what kind of small molecule, which could be potentially a drug, can actually save this patient neurons from having a bad disease signature. So we used a little chemical that came out of yeast cells that made this yeast cell grow better in this toxic protein expressing situation. And we used that small molecule in the human neuron, patient neuron, and we were able to make them better in certain signatures. This reprogramming technology was not around, let's say, before 2007. So I hope in my little dream is this platform that we came up with, the simple model system yeast and iPS neuron model system this from the patient, can help understand or discover and the drug maybe that could help patients. We've been going around doing fundraising, and these patients are involved in this fundraising because they really do want a cure, and the only hope for them is for us, the scientists, to come up with understanding of these diseases and come up with the cure. So just experiencing that firsthand from not so far from a patient point of view really keeps me work hard on the problems that we have, and hoping that this could be facilitating the research process. So it's a lot of responsibility, but also could be very fulfilling.